All right, so we're going to start part B, which is going to talk a little bit about the structural classification of neurons and action potentials. Okay, so we just finished talking about the functional classifications of neurons, and if we go back a little bit, we can see that some of these functional groups, remember, were things like the motor neurons that carry impulses from the nervous system to organs and muscle. Interneurons are found within the central nervous system, and they integrate the signals coming in from sensory neurons, allow something to happen, integrate it, decide what to do with that signal, and then going to send out some kind of response through motor neurons. Okay, so when we talk about the functional classification, we have to look at sensory. Okay, so we've got sensory neurons that bring information from the environment. Okay, then it's processed, through these interneurons. Okay, and then some kind of response goes out through the motor neuron. So you've got a sensory neuron, an interneuron, and a motor neuron, and those are classified by their functions, what they do. Okay. Now in this part, let's talk a little bit about, this is starting part B, let's talk a little bit about their structural classification. And when we look at neurons, there's three kind of main types of neurons, and they are classified structurally based on the number of processes that come off of the cell body. Okay, and when we talk about processes, we're talking about dendrites and axons. Okay, so when we look at something called a multipolar neuron, it has many extensions that come off of the cell body. Okay, it's going to have many dendrites coming off the cell body, and then one axon. Okay, all motor and interneurons are multipolar. So the majority of neurons in the body are multipolar. It is the most common structural type, and this is kind of what it looks like. So if you look at this, here's the cell body, and when you look at the projections that are coming off, you can see, here's 1, 2, 3, 4, 5, 6, 7, 8, 9, and then here it would be like 10. Here's the axon. So there's lots and lots of different extensions that are coming off of the cell body. So there's multiple extensions coming off the cell body. That is what we call a multipolar neuron. Okay. There's another type of neuron that is called bipolar neuron. Okay, bipolar, bi meaning two, which means that there's only going to be one dendrite and one axon that's going to come off of the cell body. Not found in a lot of places in the body. Mostly they're found in sense organs like the nose and the eyes. Pretty rare in adults. You'll find these in the eye, in the retina, that detects light. Okay, and this is kind of what they look like. If you can kind of see here, you can see that there's only two extensions that are coming off. So here's the cell body. So you've only got one dendrite and you've only got one axon. Okay, so that's just two extensions coming off of the cell body, which makes it bi. Bi means two. Okay. Then we have another one, another type of neuron, and there's only one extension coming off the cell body. So that would be called a unipolar neuron. Okay, they have a really short single process leaving the cell body. Sensory neurons found in the peripheral nervous system ganglia. They conduct impulses both towards and away from cell bodies. This particular type of neuron is really good at sending signals long distances. Okay, and when you look at this neuron, this is what it looks like. Okay, you can see here's the cell body, and there's only one little extension there. So this is unipolar. So it's got one little extension that's got this really long axon. Okay, and like I said, this is really good at sending signals throughout the body. Okay, getting a signal from one place to another. Okay, so you can see kind of this is the dendritic end. Here's the axonal end with the axon terminal, and then it's just got this one little single process that leads into both the long dendrite and the long axon. Okay, or I should say these are the dendrites down here. This is the long axon. Okay, and like I said, they're found really in the sensory neurons found in the peripheral nervous ganglia. Okay, so they're going to be found in eyes, ears, anything that has a kind of a sensory neuron. Okay. And we'll talk more about that, or I'll give you examples of that when we get to it. Okay. Now, also in terms of functional properties is really what it does. So when we talked about muscle, do you remember we said that muscle was irritable? It had conductivity to it. Same thing with neurons. Irritable means it's able to respond to a stimuli, and it can convert that stimuli into a nerve impulse, which we would call an action potential, just like muscle was able to do. Okay, it can respond to a stimuli, and then it can cause muscle contraction. Nerves don't really contract. They can actually send a signal to another nerve or to another muscle, so forth. Okay, so they can send signals to each other and to other effector cells like muscles and glands. They are able to conduct these signals for long distances, and so they have what we call conductivity. Okay, so they have the ability to transmit the impulse from other neurons, muscles, and glands. Okay, so they can conduct these impulses for long distances. Okay, and they can conduct them between one neuron and another neuron, or they can conduct them between a neuron and muscles and glands. Now, how do they do this? Okay, well, they conduct these signals electrically. Okay, and the nice thing is about this is that we've already talked about all this when we talked about muscle, so this really is going to be a review for you all. Hopefully you kind of understood it from muscles. If you didn't, here's your second chance of learning all about it. Okay, because really the same process is happening in nerve tissue as it does in muscle tissue in terms of calcium, voltage gated calcium channels and vesicles and neurotransmitters and synaptic cleft and sodium ligand gated channels, and causing an action potential, causing the membrane potential to flip. Remember, you're usually positive outside, negative inside. When you flip that potential, you get an action being able to happen, which is what we call an action potential. Okay, so let's kind of review this. Just hopefully this won't take too long because, like I said, we've already talked about all of this. This is why I kind of hammered it pretty hard in muscles, because I knew we'd be learning the same process in nervous tissue, which makes it kind of nice as a review. Okay, so it says electrical conditions of a resting neuron's membrane. Okay, so when the membrane is at rest, what does it look like? Okay, so if we were to draw our phospholipid bilayer, so here's our membrane, here's our double layer, our membrane. Remember, typically at rest, we are positive outside and we're negative inside. And remember, when I say negative, I don't mean like there's lots and lots of negative charges. I mean that we're more positive outside than we are inside. Okay, because remember, there's still potassium in here. But let's say if I draw like 2 potassium, but let's say I draw out here like 4 potassium. So we're more positive outside the cell than inside the cell. Okay. All right, and sodium works the same way too. So I'm just using potassium here. Okay, but potassium, there's more potassium outside the cell, so there's more positives outside the cell than there are inside, which makes this outside more positive than the inside. So we call this positive and this negative just because there is a difference in charges, meaning there's more positive outside the cell than inside the cell. Okay, so there's fewer positive ions inside. So here we go, fewer positives inside the neuron's plasma membrane than outside, that work just like this in muscle cells. Okay, so potassium is the major positive ion inside the cell. Okay, sodium is the major positive ion outside the cell. And why is that? Why is this way? Well, do you remember also in this membrane are these special proteins, and they pump what in and out? They pump, remember, 2 potassium in, and they pump 3 sodium out. Okay, so there's more sodium outside the cell than there is inside the cell. There's more potassium inside the cell than outside the cell because we're pumping in potassium and we're pumping out sodium. Okay, so this has to do with the sodium-potassium pumps. That's why this happens. Okay, the polarized membrane is more permeable to potassium than to sodium. Well, why is this? Well, do you remember also in the membrane were these leak channels, and there's more leak channels for potassium than there are sodium. And remember, leak channels are always open. They don't ever close. They're always open. And there's more leak channels letting potassium out than letting sodium in. Okay, so the membrane's more permeable to potassium just because there's more leak channels that are letting potassium leak out. Okay, you know, let's say for every 2 potassium leak channels, there's only 1 sodium leak channel. Okay, so there's going to be lots more potassium going out than sodium coming in. Okay, and so this also, because you're losing more positives inside the cell than you're gaining, this also makes it very, very positive outside the cell as compared to inside. So it says as long as the inside of the membrane is more negative, and that just means fewer positive ions than the outside, the cell remains inactive. Okay, so as long as we have positive out and negative in, okay, this is going to remain inactive. This is what we call resting membrane potential, meaning that you're going to have positive outside, negative inside, less positive inside, okay? And so you're going to have what we call rest. When you flip the charge, when you cause the inside to become more positive than the outside, that is what we call an action potential, okay? And that's when you can allow something to happen, okay? And we kind of, like I said, we've already kind of talked about that when we talked about muscles. Okay, so let's take a look at this, okay? So first, resting membrane potential. So remember, we have more positive outside than we do inside. So even though there's potassium inside, there's just not as many positives as there are outside, okay? So the inside tends to be slightly negative. So in the resting state, the external face of the membrane is slightly positive, its internal face is slightly negative. The chief extracellular ion is potassium, whereas the chief intracellular ion is potassium. So extracellular means just outside the cell, intracellular means inside the cell. Okay, the membrane is more permeable to potassium ions, which exit the resting cell in fewer positive ions inside at rest. Okay, so what I'm saying it's more permeable is because there's more leak channels for potassium in the membrane than there is sodium. Okay, so it's more permeable to it just because it can leak out faster. Okay. So action potential, initiation, and generation. So a stimulus changes the permeability of the neuron's membrane to sodium ions. So what happens is voltage-gated sodium ions will swing open and it allows sodium to rush into the cell. And when that happens, you're going to get lots and lots of positive ions rushing into the cell, and now you're going to get the cell becoming more positive inside. Okay, so it's becoming less negative. And when that happens, we call that depolarization. Depolarization, and I didn't use this word a lot during muscles because I didn't want to, I didn't want to confuse you all too much. But depolarization, when you have a depolarization of membranes, it's the same thing as saying action potential. And a depolarization means that you're flooding the inside with sodium ions, making the inside very, very positive. And when that happens, it depolarizes, meaning that it becomes more positive than negative. And so you're depolarizing it, okay? So you are getting what we call an action potential, okay? And this happens when you get a rush of sodium ions inside the cell. And you can kind of see this here, okay? So you get some kind of stimulus happening and it causes this flip in charge where you were positive outside, now you're flipping it and you're getting positive inside, okay, because sodium is starting to rush in. And you're going to get this wave of depolarization down the neuron, okay? So the stimulus initiates local depolarization. So a stimulus changes the permeability of a local kind of a patch of membrane, and the sodium ions diffuse rapidly into the cell. This changes the polarity of the membrane, so the inside becomes more positive and the outside more negative at that site. And as this happens, it's going to cause more voltage-gated channels to open and more sodium is going to rush in. Going to cause more, it's called propagating. So as it's moving down the membrane, it's going to flip these charges as it moves down, okay? And that's how this stimulus gets moved down the axon of the nerve cell, okay? So a graded potential, so localized depolarization, exists where the inside of the membrane is more positive than the outside. So we're just flipping the charge. We've talked about that a lot. If the stimulus is strong enough and sodium influx is great enough, local depolarization activates the neuron to conduct an action potential, which we call a nerve impulse. So before an action potential was a muscle contraction, in a nerve cell it's called a nerve impulse. Okay, so but same concepts happening in the nerve as it did in the muscle. Okay, so you can kind of see this wave. Now we're flipping the charges from, we were negative inside, positive outside, now we have flipped it. So the darker color here is where you're getting this depolarization, where you're getting these channels are opening, these voltage-gated sodium channels are opening, and now you're getting a flood in of sodium. And that's going to propagate all the way down. If this stimulus is strong enough, it's going to propagate or move all the way down the nerve, okay? So if enough sodium enters the cell, the action potential nerve impulse starts and is propagated over the entire axon. And it's interesting in muscle cells, it's called an all or none response. It means that it has to reach a certain threshold in order to get this process to happen. If you don't reach threshold, the nerve impulse will not happen. Okay, so it's an all or none. Once that the nerve impulse does happen, it'll propagate it all the way down the axon. Okay, so all or none response means that the nerve impulse either is propagated or not. You can't have any gray area. Either it does happen or it does not. Okay, so that's what we call all or none response. Fibers within the myelin sheaths conduct nerve impulses more quickly. So do you remember we talked about nerves. So here's the cell body, here's the nucleus, here's the axon terminal, and then we had these myelin sheaths, these Schwann cells, okay? And they're kind of fat layers, okay? They help to insulate. If there's myelin sheaths around the axon, what will happen is that the impulse will kind of jump. It will move faster if it's myelinated, okay? Because it doesn't have to move all the way down the axon it can actually jump from node of Ranvier to node of Ranvier, okay? And it will conduct it much faster when it's myelinated like this, okay? It's kind of like insulating a wire because you don't lose any ions. The ions aren't lost all the way down, so you can actually kind of keep the ions where they're supposed to be when you kind of line them with this myelin sheath here, okay? That's kind of how I like to think of it. Might not exactly happen that way, but I kind of like to think it's insulated so the ions can't, they kind of get insulated in here and it allows it to kind of jump from node of Ranvier to node of Ranvier without having to go down the entire axon, okay? So it's going to keep happening. So propagation of the action potential, depolarization of the first membrane patch, remember, which was up here, okay, causes permeability changes in the adjacent membrane. So you're going to get some permeability changes here, and then you get them here, and then you get them here, and it keeps going until you reach the end of the neuron, which you're going to get to an axon terminal. So the action potential propagates rapidly along the entire length of the membrane, like dominoes falling one after another, or the crowd doing the wave. Remember, and I talked a little bit about that domino. I don't know if I mentioned it in your class, maybe it was the other class, but it's kind of like stacking up dominoes, and when you hit one domino, if it's all or nothing, all of the dominoes will fall down. And that's kind of what's happening here, where it gets propagated. If it's strong enough, it'll open up sodium-gated channels and it'll propagate all the way down the neuron, okay? Now, once you get this depolarization, you have to reset everything. And when you reset all the charges so that you have positive outside again and negative inside, that's called repolarization. And it's a time when the membrane permeability starts to flip again so that it can go back to resting membrane potential. So membrane permeability changes again, becoming impermeable to sodium ions and then permeable to potassium, okay? Potassium ions rapidly diffuse out of the neuron, repolarizing the membrane. So all those leak channels then kind of take over, and there's also gated channels that will open up and allow potassium to rush out of the cell. And that allows the cell to become slightly less positive because you're losing a bunch of potassium ions. So the outside becomes very positive and the inside becomes less positive, or what we call negative, okay? Repolarization involves restoring the inside of the membrane to a negative charge and the outer surface to positive. So just remember, depolarization is flipping the charge and repolarization is resetting everything. So I like to think repolarization, resetting. So it allows it to reset it back to positive inside, negative outside. Okay, and you can kind of see that here. So when we repolarize either muscle or a nerve, it is going to cause now potassium to rush out of the cell. And when you get this potassium rushing out, you're losing more positives, and it's going to become more positive outside, and you're going to become less positive inside because you're going to be losing a bunch of potassium. Okay, so leak channels open up, and then there's also voltage-gated potassium channels, that open up and allow potassium to leave the cell in great quantity. And because you're losing so much positive, you become slightly negative inside or less positive inside. Okay, so potassium ions diffuse out of the cell as the membrane permeability changes again, restoring the negative charge to the inside of the membrane and the positive charge to the outside. Repolarization occurs in the same direction as depolarization, meaning that it occurred in this direction. Our impulse was moving this way. So repolarization is also going to move this direction. Okay, so they both move in the same direction. That's all that's saying. Okay, so repolarization. So the initial conditions of sodium and potassium ions are restored, okay, and it's also helped with the sodium-potassium pump. So sodium and potassium pumps are going to start using ATP to pump sodium out and potassium in, okay, so there's 3 sodiums pumped out for every 2 potassiums that are pumped in, okay, and this will happen until repolarization is complete. So when the nerve is back at resting, where you have positive outside, negative inside, okay? And a nerve cannot conduct another signal until it is repolarized, okay? Until it goes back to resting and everything is reset, it will not be able to conduct another impulse, which is a little different in muscle. Remember we had that unfused tetanus where it doesn't have to completely relax before it gets an impulse again. Sorry, in nerve cells it does have to completely relax before it can get another impulse, okay? So here is the sodium-potassium pump, okay? And do you see how this is a little bit bigger? The arrow for potassium is a little bit bigger, and like I said, because this is a little bit bigger arrow, and that's because there's more potassium leak channels. It's an A, sorry, channels. And do you see how this is a little bit smaller? This arrow is a little smaller, and that's just because there's less sodium. This is potassium, sodium leak channels. Okay, so more potassium is going to diffuse out, okay, than sodium diffusing in. And then remember, we're losing 3 sodiums outside the cell and only gaining 2 back in. Okay, so the outside is becoming much more positive than the inside is. Okay, so this is going to be slightly negative, meaning it's just not as positive as the outside. Okay. Okay, so let's talk a little bit about the transmission now. And like I said, this is going to occur just like it did in muscle. So all of this is not new, okay? So transmission of the signal at the synapse. Okay, it says step 1. When the action potential reaches the axon terminal, so that action potential that we just talked about, when it reaches the axon terminal, so the end of the axon, the electrical charge opens calcium channels. And we've learned that. Remember, these are voltage-gated calcium channels. So voltage-gated calcium channels, okay? Those calcium channels are going to open up, and I bet you I don't even have to keep going with notes. I bet you guys are going to be able to figure out what happens next. So when calcium rushes into the axon terminal, what happens? What is going to happen before I even move to the next slide? Okay? So if you remember from muscle, okay, so when we get a signal coming down, it's going to cause these voltage-gated calcium channels to open, and it causes the vesicles that are filled with neurotransmitter to fuse with the end of the axon, okay? And when they fuse with the end of the axon, they're going to release acetylcholine into the synaptic cleft, okay? That acetylcholine will then bind to these ligand-gated channels. Remember, ligand has to have some chemical to bind to it before it will open. It's going to bind to these little ligand-gated channels, and when they bind to it, like this one you can see, do you see how it's open? It's going to be open. Now, what channel is this? What channel is this going to be down here? Okay, so when this little ligand binds to it, like you can see here, it's bound to it, it's going to let something flow into the cell. What is it going to let flow into the cell? And if you said sodium, you guys are so right. Okay, it's going to open up and allow sodium to rush into. Now, if this is a muscle, this could be another neuron. Okay, so it doesn't necessarily, when we are talking about nervous system, this could be a gland. Okay, this could be muscle. This could be another nerve cell. Okay, it could be another neuron. So it could be any one of these. Could be this cell right here. Okay, but the same thing's happening. Okay, where you're going to get these vesicles fusing, and it's going to release acetylcholine. Acetylcholine is going to act as a ligand. It's going to bind to this channel, open it up, and now sodium is going to rush in. And when we get sodium rushing in, when you get a rushing in of a bunch of positives, what happens? Yes, you flip charges, you have an action potential, and you now have gone positive inside and then negative outside. Okay, so you flip the charge. So all of this is going to be a review. Look at this. Okay, so calcium in turn causes tiny vesicles containing the neurotransmitter to fuse with the axonal membrane, okay, and you can kind of see that there. Okay, so these are going to be sodium channels here. Okay, here's the little acetylcholine. Okay, it is bound to the gated channel. Okay, this is the ligand gated channel, right? And when that happens, the entire of the calcium the entry of calcium into the axon terminal causes pore-like openings to form, releasing the neurotransmitter into the synaptic cleft. Okay, so we know that. Okay, it's going to end up binding. Okay, so neurotransmitter is released into the synaptic cleft. Okay, the neurotransmitter molecules diffuse across the synaptic cleft and bind to receptors on the membrane of the next neuron. Okay, and like I said, it does not necessarily have to be a neuron. Okay, it could be a neuron, it could be a muscle cell, it could be a gland. Okay, it doesn't have to be a neuron. Okay, it could be any one of those types of cells. Okay, and here's our picture. Okay, so neurotransmitter binds with the receptors. Okay, and it's going to open them up. And when it opens up, okay, if enough neurotransmitter is released, a graded potential will be generated. Okay, meaning that you're going to get those ligand, that acetylcholine, bound to the gates on the other side, and they're going to open up. And then eventually it's going to flood the cell with sodium. Okay, so eventually an action potential nerve impulse will occur in the neuron beyond the synapse, okay, or muscle cell or gland, okay, if you have enough there. All right, you can get, now see, you can see here's sodium rushing in. So here's our neurotransmitter. It's going to cause this to open up, and it's going to cause in a rush of sodium. Okay, so ion channels open, and it's a sodium gated channel. Okay, just like muscles. This is all review. Okay, so the electrical changes prompted by the neurotransmitter binding are brief. The neurotransmitter is quickly removed from the synapse either by uptake or by enzymatic action. And if you remember, there is an enzyme that breaks down acetylcholine. It's called acetylcholinesterase, and that will break down acetylcholine so that it stops this process from happening. Okay, so transmission of an impulse is electrochemical. Transmission down the neuron is electron because you're flipping charges, and then transmission to the next neuron or muscle or gland is chemical, and that's used by the neurotransmitter. So you're going from an electrical signal to a chemical signal, and then ultimately back to an electrical signal, because you're going to cause an action potential to happen in that cell of the next cell. Okay, all right, which, like I said, could be another neuron, it could be a muscle cell, or it could be a gland. All right, so neurotransmitter is broken down, and then and it's released. So you can break this down. There's an enzyme that does this called acetylcholinesterase, and then it's going to break down. It's going to release from this and cause this channel to shut. And now sodium can't get into the cell anymore, and so now your action potential is going to stop. And then you're going to get repolarization happening, where you're going to get potassium channels opening up and allowing this to reset itself. Okay. Okay, so let's see if I can, see if we can watch this.
Our brains are constantly processing information, which requires communication between billions of nerve cells. Signals are passed from a sending neuron to a receiving neuron at a junction called a synapse. An action potential in the sending neuron travels down the axon until it reaches a synaptic terminal. The narrow gap between the synaptic terminal and the receiving neuron is called the synaptic cleft. The synaptic terminal of a sending neuron contains numerous vesicles filled with neurotransmitters, chemicals that carry information across the synaptic cleft. When an action potential reaches the synaptic terminal, the vesicles fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters affect the receiving neuron, changing the distribution of charge across its membrane. Let's take a closer look. An action potential is propagated down an axon by the opening and closing of sodium and potassium channels. When an action potential arrives at the synaptic terminal, it causes the opening of calcium channels, shown in green. Calcium ions enter the synaptic terminal through the calcium channels. Calcium ions bind to the vesicles containing neurotransmitters. This causes the vesicles to fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters diffuse across the synaptic cleft and bind to receptors in the plasma membrane of the receiving neuron. Here, the receptors are ion channels that open. ions move across the membrane, changing the distribution of charge across the membrane. The neurotransmitters are quickly removed from the synaptic cleft, ending their effect on the receiving neuron. A single neuron can receive signals from many sending neurons. The signals arriving from the axon on the left are excitatory. They make the receiving neuron more likely to generate an action potential, as indicated by the green glows. Signals from the axon on the right are inhibitory. They make the receiving neuron less likely to generate an action potential, as indicated by the red glows. The two sets of signals cancel each other out, and no action potential is generated. Now, only excitatory signals are transmitted, and the receiving neuron generates an action potential down its axon. The signals that pass between these and countless other neurons constitute our thoughts and coordinate our activities.
Okay, so that was the end of part B. So hopefully that was just a brief review of what we talked about in muscles. So that ends part B.